Class 1, Basic Anatomy and Methods. Neurons. In the last class, we talked about why we should care about the brain. This class is about the how. That is, what are the methods that we can use to study the brain? What should we focus on? There are many ways to study the brain. It's one of the most complex things known in the entire universe, and so there are different levels at which we can study it. That is, there are different levels of details in terms of anatomy. Let's start with the smaller units that we need to understand. Neurons. They are some of the most important cells in the brain. Why? Because they send signals to each other. A typical neuron looks like this. The soma is just the body of the neuron. The axon is the output channel where it sends the signals, and the dendrites are where it takes the input from other neurons, often from many other neurons. Essentially, the neuron accumulates signals from other neurons through the dendrites. When the accumulated input of these signals reaches a certain threshold, that neuron is said to fire, which means that it sends out a signal through the axon to other neurons. These signals are also called action potentials. Action potentials can be measured as a change in the surrounding electric field. That is, these are electric signals. However, between neurons there are gaps called synapses. The electric signal does not pass through these gaps directly. Instead, neurons communicate with each other chemically through the synapses. The details of how synaptic communications work can be very complicated, but they're not important at this point. What you need to know is that essentially, neurons are simple electrochemical signaling devices. This is interesting because it means that if you change the chemical content of the blood inside the brain by, for example, ingesting drugs, you change the behavior of the neurons themselves. Normally, drugs don't go inside of the brain because they cannot pass through what is called the blood-brain barrier. Because the brain is so important, there is a mechanism to stop most substances in the blood of the body from entering the brain. But some drugs are psychoactive. They have the right molecular structures to go through this blood-brain barrier and influence how the brain functions. But it's not all just chemistry. If we measure the electric fields around a neuron, you can measure its activity too. This is why we can put electrodes invasively into animals' brains to measure the activity of single neurons. We can also put electrodes onto human scalps to detect the weak leakage of these signals from the neurons. This is the basis of the EEG, or the electroencephalogram. We'll talk about this more later in class. There are many different types of neurons. Some have bigger bodies than others, and they have different firing properties. In other words, some fire at different rhythms or frequencies, which is measured by the standard unit of hertz, otherwise known as spikes per second. And then there are other brain cells too, which don't carry these signals. For example, there are glial cells. They are important and carry out essential housekeeping work, like cleaning up the chemical content surrounding neurons. But for now, let's focus on the neurons. They are the main ingredients that enable the brain to compute stuff. Brains are full of these neurons. In the human brain, there are tens of billions of them.